today I'll be talking about this method SOFA, which is a preprint. So the title is Semi-Supervised Ohmic Factor Analysis, disentangles known and latent sources of variations in multi-omic data. So very related to MOFA from some of the same authors, and with a little bit of a difference, which I'll discuss today. So the DOI here. So as an overview, basically, so as I mentioned, it's very similar to MOFA, where it's a mathematical technique where you essentially get these factors that are incorporating data across a bunch of different omics. So you have the same set of samples, and then you have different measurements on those samples. So that could be very diverse things. It doesn't have to be measurements of genes or transcripts. It can also be different scores they give a lot of different examples, but it can be really any type of quantitative information. So mathematically, it's it's a linear method, so sort of similar to PCA in some sense. Really, it's using a low-rank matrix decomposition, and we'll get into that sort of in the next figure. So I'm I guess we've seen MOFA before, so I'm sort of assuming some knowledge of MOFA, but the thing that makes it a little bit different is these guiding variables. So in the method, you can sort of specify sample level information like sex or age, other variables like that. And the idea behind that is that these factors are sort of finding different aspects of the biology, and you can sort of guide certain factors to find the variation related to those sample level covariates. And then once you do that, that basically regresses out those known sources of variation, so that when you have unguided factors, those focus on basically novel biology aspects that you weren't necessarily expecting. And so that's sort of a way to guide the method to find things that are new versus already known. So it is an open source Python package, and I'll get more into the software sort of towards the end of this. So the figure one really focuses on the method itself. So in A, essentially they're showing this low-rank matrix decomposition. And so you have basically the information I guess we'll start on the left, where you have the different features and the different views. So these views, there's a lot of ways to define these views. So with MFA or with MOFA, not SOFA, previously we've done views sort of like spatial domains. So the features can actually be shared across the views, but they don't necessarily have to be. So you could have views that are one view is transcriptomics, the next one's proteomics, so they don't share features whatsoever. And then you have your different samples as the rows in this case. So you can supply as many views as you want, as much data that you have about your samples. And then the SOFA method is breaking that down into a set of factors and loadings. So importantly, the factors are broken up so basically you have your factor scores where each sample has a score. So in this middle piece here. And importantly, it's broken up into guided and unguided. So like I mentioned, the guided are you provide this set of sample level variables that you want to guide those factors. So that's up to you. And then unguided factors will just find sort of the remaining variation and discover potentially novel biology. And then so you have those factors which have sample level values, and then you have the loadings which give you a value for every view and for every feature. So you can see certain features are going to contribute more to different factors, and potentially certain views may be more relevant in the factor, which we'll get into with some of the example data sets that they cover. There's some mathematical details here, they do actually sort of encourage sparsity in the loadings. So I forget if that's done with a L1 penalty or something similar. I can't remember the exact details there, but sparsity just encourages you to get sort of the most relevant information. And they mentioned that sort of on the very right here that you can have loadings that are sort of specific to one view. Maybe only one view is potentially relevant, whereas other ones can be spread more evenly across views, either all views or just a subset. But this is the basic mathematical model here that's being used in SOFA. So very similar to MOFA. In the remainder of that first figure, they show basically more details about the guided variables. So I mentioned that you can have the guiding variables can be any type of variable. So that can be continuous measurements. It can be categorical data, including just binary, 2 categories, binary. So they show some examples in panel B here of essentially the point that they make here is that the guiding factors are sort of approximately linear with the original guiding variables. So like the idea is that they're regressing out the contribution to the latent space. So all of the biology that you get once you regress that out are just sort of new things that are not predicted by your input variables, your input guiding variables. So, and then I sort of stuffed this on the right of the slide here, but panel C is sort of hinting at sort of downstream applications that you can do with once you get these factors. So they show a panel where you have multiple factors and you're seeing how different clusters basically separate based on values of the factor. So those clusters could be different aspects of the biology. For example, in this panel here, you can see values of the factor actually separate predictions of survival in cancer-related analyses. So that can be sort of one biological aspect that's being captured by the factors. Of course, since you have the loadings for different features and different views, you can rank the features by the loadings. So you can get the features that are contributing most, both in the positive and the negative direction for a given factor. So those are just like the most relevant features. And then once you have those lists of features, you can also do like things like Go enrichment analysis so that you can get the top pathways related both in the positive and negative direction for the factors. So these are things that we've sort of done already a little bit with MOFA, but with the guiding variables, it can be a little bit maybe more precise. So here I'm sort of deviating a little bit from the organization of the paper, but I wanted to, they made this point sort of in several places in the paper where they're showing how the guiding variables actually do like effectively regress out the known effects. And so they do a good job of sort of, I guess regressing out the things that we already know about. And so they make this point in different mathematical ways. Sort of this panel A on the top right, I guess I should mention that this data here is talking about cancer data from the Cancer Genome Atlas project. And so panel A is colored by the different subtypes of cancer. And importantly, this model is guided by they use as a guiding variable the cancer subtype. So you would expect separation in something like a t-SNE. And you do indeed see that separation, that separation is happening because they specify this cancer subtype as a guiding variable. So in contrast, panel C is showing like an unguided version where they don't really inform the model, or they don't provide as a guiding variable the cancer subtype. So the model doesn't naturally separate that out as one of the most important sources of variation. So that's one way, I guess, the guidance is affecting the model. So the guidance, like naturally, the model is not going to necessarily pick up variables that you think are important, like age or sex or cancer subtype. But if you want to regress out this effect specifically, guiding variables are effective at doing that. And the panel A I think this was from a supplementary figure on the top right, they make this point in a similar way where these 5 factors are guiding factors, and then the color. So they have the different cancer subtypes, and then the factor values separate out. I forget how they did this actually. I think they separated out I'm blanking a little bit, but basically the factor sort of separates out the guiding variable, since these are guiding factors. So it's just different ways to show that the guiding variables do or regress effects. In panel E here, they did sort of a clustering, and this was a little bit more complex. They had some methods in here describing this in more detail. But the general idea is that when you provided the guided variable of the cancer subtype, like that was very high in ARI when you used like the clustering data from this compared to the categories of the cancer subtypes. Whereas if you didn't guide the model, that there wasn't a powerful relationship between the cancer subtype and what the model was finding. So you did really need to guide the model in order for it to separate out those cancer subtypes naturally. So this is going more into some of the downstream analyses from this Cancer Genome Atlas, that one of the applications that they mentioned in this paper. So I guess I touched on this a little bit, but panel C sort of goes into this. They found this factor 10 that they noted was related to survival in terms of cancer outcomes over time. And it did significantly separate survival based on these plots on the left, both in terms of the progression-free interval and then the overall survival number of days. Very similar was on this panel B on the right, where the hazard ratio captures something related. Basically, the higher the value here, the worse the outcome for survival. And so they have these relationships with every single factor on the x-axis. So they noted like factor 3 was also one of the important factors for. I think they made the point that the microRNA here in ovarian cancer were sort of well known to be important or very relevant in cancer outcomes. So they have I guess I sort of jumped plots here, but this bottom plot on the right here is a variance explained plot. So this is showing the contribution of the different assays or the different views really across the different factors. So again, sort of making the point that a factor is not guaranteed to have sort of the same importance in every assay. So it might be true that the mRNA are more important in a particular factor than the methylation information, or vice versa, depending on the factor. And so, yeah, there's a lot you can do with that. Going on some of the other applications, you can also just sort of use the loadings. I mentioned this a little bit earlier, but this is a more specific example of doing this, where you have loadings from the different assays. So you have your RNA loadings and then the protein loadings, and just seeing the ones that are really isolating the ones that are highest in those loadings for the factor of interest, in this case, factor 10, which we showed was interesting in terms of the survival outcome. And then naturally, this is something we've already done sort of before, is do enrichment analyses. So I think they used Go and then the reactant in panel E here, making the point that sort of these positive loadings were related to cell proliferation, obviously very relevant in cancer, whereas the negative loadings were related to like these basically lymphocyte infiltration, these sort of like immune processes being impacted by the cancer. And so that gives you an opportunity to sort of biologically interpret some of these factors in terms of the pathways that are involved. So now I'm jumping to a different application that they have in this paper. So they covered a few different data sets. This is also cancer related. This is cancer dependency map. So I'm going to sort of maybe skip a little bit over the background of this data set and sort of focus on the findings that they pointed out were sort of most interesting here. So here we have two interesting factors, factor 10 and factor 12. So these were sort of cases where the factors were distinguishing rather than like sort of macroscopic and very broad, I guess, biological categories. These factors were capturing things that are a lot more subtle. So factor 10 here, they had different types of cancer again, and they focused on cells that were like either lung cells or not lung cells. So factor 10 did not really distinguish between whether a cell was a lung cell or not. It was actually distinguishing within the lung cells. So they had these different types they had like this small cell lung cancer and then non-small cell lung cancer. And apparently this factor was distinguishing sort of within that very specific that subtype of the lung. So it was interesting that it was finding this sort of biology here. In panel G, there was something very similar. Factor 12 was also distinguishing something pretty subtle, where they pointed out that like it wasn't distinguishing between blood cells and other cells. It was distinguishing within blood cells. So they have these B, T, and myelinate, or sort of different types of blood cells. So it was obviously we've seen a pretty profound separation based on those subtypes of the blood. So factors might capture things that are very subtle and very specific to one cell type, which is pretty interesting. Further interpreting this factor 10 that was interesting in terms of the lung cell types, part of this cancer dependency map was they include information about like different cancer subtypes and then also drug responses to the cancer. So that's sort of a focus of the project. And so basically in panel E, they have the loadings. So in the loadings, they have both the RNA measurements and then also drug response information. So that's sort of interesting, again, you can sort of provide whatever quantitative measurements of your samples that you have. So in this case, it's kind of interesting that you can integrate the drug response and RNA information sort of directly into the same factors. So they identify sort of top drugs that are interesting here, as well as the top genes. In terms of, they also looked at sort of like mutations. So there's this volcano plot in panel H here, sort of separated. So they were trying to measure the association of factor 10 with certain mutations. So that's sort of like where we get an effect size and a p-value. And so certain genes were, certain mutations in very specific genes were implicated for factor 10 specifically. So I'm not that familiar with biology of these genes, but this would be something that would be an interesting finding in terms of the cancer here. So moving on, sort of basically the software, the technical end of the paper, I guess. So as I mentioned, this is Python-based software. It seemed pretty, looking at it, it seemed pretty interesting, like easy to install. So it's just available through pib, just a Python package. So they recommend like creating an environment with like Python 3.10. I'm not sure if that'll be absolutely necessary, but this might just end up like a module, which would be pretty easy to create. I looked through and in the tutorials, they do recommend using a GPU. I'm not sure in terms of the runtime if there's any information about that, but if it's, I'm guessing since it seems very similar to MOFA, the runtime should be pretty fast. And I'm not sure we'll even need a GPU, but they do recommend it. The documentation looked pretty good, in my opinion. So they had like this website with obviously the API, the different functions that you can call, as well as like sort of documentation reproducing the analyses in the manuscript. So they sort of separate it down here by the three data sets that actually one of these I didn't cover today, but they have like the depth map, the GCA, and then the other one I didn't cover today. But yeah, reproducing everything from the manuscript. So, and it looked pretty comprehensive in terms of like comments, which we can take a look at if we're interested. But yeah, so I think this should be pretty straightforward to apply in like our workflows. So I guess in conclusion, SOFA is very similar to MOFA, but sort of extends it in terms of allowing you to specify guiding variables so that when you guide the model, you can sort of limit the unguided factors to find sources of variation that are novel with respect to those guiding variables, at least. And it's a very useful method in terms of being able to integrate information across a lot of different assays and measurements, any sort of quantitative information, as long as you have the information for the same set of samples. And then there's a lot of downstream applications that you can do once you have these factors to sort of interpret them biologically. So different ways you can do that is you can look at variants explained to see what are the relevant views being implicated, what are the relevant factors. And then you can sort of, we've mentioned checking the top weighted features for different views. And once you have those, you can do things like go enrichment to find different pathways that are implicated. And then depending on like what you're studying, so we mentioned with cancer, there's like survival analysis. So there's probably a lot of different, depending on the domain you're studying, there's different interpretations that might be interesting for looking at the factors.